Hello students, welcome back to the channel. In this video, we are going to give you a quick revision of this chapter. I hope this video will be helpful for you. Now let us start. So see beta, unicellular organisms are capable of independent existence and they are performing the essential function of life. Anything less than a complete structure of cell does not ensure independent living. Hence, cell is fundamental structural and functional unit of all living organisms. Question area, how do they frame? They frame this way. Anything less than a complete structure of cell ensure independent living. So this is wrong. It does not ensure. This way they will frame it from here on. Now you know very well who discovered cell. We say Robert Hooke, but he discovered cork. The cork is nothing, you know very well, it is a dead cell. The first living cell was studied by Leeuwenhoek. Anton von Leeuwenhoek is the first who saw and described the living cell. The first scientist. Second is Robert Brown, who has discovered the nucleus. And later on, invention of microscope and its improvement leading to the electron microscope revealed all the structure in detail. We were able to do it. So I think so this much is clear to you. Now cell theory. Here also you have to learn because they are asking the question. In 1838, Schilden, he is a German botanist. Schwann is a zoologist. Schwann is a zoologist, okay? So what he observed plant and he found that all plants are composed of different kinds of cells. Schwann in 1839, next year, he was a British zoologist. So Schilden was German botanist and it was British zoologist and he studied animals. And he found that cell has a thin outer layer, which is today now known as plasma membrane. And he also concluded, based on these studies, that presence of cell wall is unique characteristic of the plant. So question, who studied both plants and animals? Answer will be Schwann. Who told that cell wall is unique feature of the plant? Again, answer will be Schwann. Though he's a zoologist. He studied animal first and he told animals are having cell membrane. Later he studied plant also and he told plants are having the outer membrane that is known as cell wall. On this, Schwann proposed the hypothesis. Please, Schwann proposed the hypothesis that the bodies of animals and plants are composed of cell and products. So Schwann only proposed the hypothesis. Later on, they together have formulated the theory. Schilden and Schwann together formulated the theory. And this theory, what it was saying, all living organisms are composed of cell and cell product. But this theory was not able to tell how come new cell is coming. For this, Rudolf Virchow in 1855, he first explained that new cell arise from pre-existing cell. New cell are formed from pre-existing cell and he gave the name as omnis cellula e cellula. Is it clear? So he is the one who finally modified the cell theory and he gave these two concepts. So I hope up till here you are able to follow better. Now let us see overview of a cell. So if you see beta, we are taking one example of the plant like an onion cell we are taking. And for animal cell, we are taking the cheek cell. The human cheek cell, we are taking it. So in onion cell, what we are finding, they have a distinct cell wall as their outer boundary. And just within it is a cell membrane. But in human cheek, what we are finding, it is having outer membrane as a delimiting structure of the cell. Inside this, you are finding a dense membrane-bound organelle known as nucleus. And what does nucleus contain? It contains chromosomes, which is nothing, genetic material, DNA. Chromosomes are made up of proteins and DNA is a genetic material. Eukaryotes are those cells which are having membrane-bound nucleus and prokaryotes are those which does not have membrane-bound nucleus. You know this definition. So cells which have membrane-bound nucleus are known as eukaryotes, whereas cells which lack membrane-bound nucleus, they are known as prokaryotes. But whether prokaryotes or eukaryotes, they are having one common arena for all the cellular activities. That is known as cytoplasm. So cytoplasm is the main arena for all cellular activities, whether in plants or animal cell, and whether both prokaryotes or eukaryotes, this is present, which is occupying the volume of the cell. Again, question area. Main arena for cellular metabolism is cytoplasm. Present in both prokaryotes, eukaryotes, whether it's plant cells or animal cells. Chemical reactions are occurring to keep the cell in the living state. Living state, we say it is unequilibrium state, where the cell is trying to prevent itself to fall from equilibrium. If the moment equilibrium comes, the person, we call it as dead person now. So living state is a state where they are trying to maintain unequilibrium. Other organelle, if you see, which are membrane bound. Membrane bound organelle is your ER, Golgi, lysosomes, mitochondria, microbodies, they have mentioned here, vacuoles. Prokaryotic cell, you know very well, they don't have membrane bound organelles. Ribosome is membraneless. Ribosome is membraneless. It is non-membrane bound organelle found in both prokaryote and eukaryote. And you know, in case of prokaryote, it is 70S ribosome. In case of eukaryote, it is 80S ribosome. In case of eukaryote, and here this is in case of prokaryote. Within the cell, ribosomes are found not only in the cytoplasm, they are present in two organelle, chloroplast and mitochondria, and as well as on rough ER also. So if they are present in the chloroplast and mitochondria, here the ribosome is of 70S. In mitochondria and chloroplast, ribosome is of 70S. So this much is clear. The one cell organelle which is membraneless is ribosome. We have studied where are they present. Another cell organelle which is membraneless is centrosome. But centrosomes are present only in animal cell. They contain another membrane, non-membrane bound cell organelle, and this helps in cell division. So this much I think so outlook is clear. Now let me make the mind map to explain you. Just one minute. So you can make this way cell. We are talking right now eukaryotic cell. Cell wall. It means in the plant and fungus. Eukaryote we are talking about. Then is your protoplast. Protoplast means cell membrane and protoplasm. Protoplasm means cytoplasm and nucleoplasm. Nucleoplasm, you know very well, it is present in the nucleus. Cytoplasm is having organelle and cytosol, liquid part. If you remove the organelle, what is left? That is known as cytosol. Organelle, which are single membrane bound, single membrane. Single membrane, you can write ER, Golgi, lysosome, vacuole, and microbodies. Then double membrane. Double membrane, we will write mitochondria, chloroplast. Then we will write membraneless. And membraneless example, ribosome and centrosome. And when we write centrosome, that time we will focus on animal cell. So this is the mind map for the entire thing, what we studied till now. Mycoplasma is the smallest cell. Please learn this much. Bacteria is this much. Now question, listen how the question they are asking. Bacteria is given 3 to 5 na, and see here, typical bacteria is given 1 to 2. Typical bacteria is given 1 to 2 and their bacteria is given 3 to 5. So please remember when they ask in the question typical bacteria, then answer will go 1 to 2. If they ask only bacteria, then it will be 3 to 5. You know mycoplasma, what they are giving, beta? Mycoplasma they are giving us, here NCERT is telling us it is only 0.3 in length. And you know PPLO, PPLO, this is point 1. So my PPLO is a category in mycoplasma only. PPLO full form you can write down pleuropneumonia-like organism. It is pleuropneumonia-like organism. It is given here. PPLO means pleuropneumonia-like organism. Is it clear? Though they are saying mycoplasma is the smallest, but and they added the word and. So PPLO in the diagram they have mentioned it's more smallest than the mycoplasma. But in the size when they give, then only we'll select the PPLO, otherwise mycoplasma is the smallest cell. So now question, if you see here in the diagram, typical eukaryotic cell is 10 to 20. The question, typical eukaryotic cell is 10 times to the typical bacteria. Yes, it is 10 times to the typical bacteria. Is it clear? It is 10 times to the typical because it's 1 to 2, so it is 10 to 20. So typical word, you have to remember it. Virus this much milli in the size. So size are different. Remember this. Clear, beta? Now see the shape. If you see in the diagram, they have given the shape. RBC, round and biconcave, WBC, amoeboid. Your columnar epithelium is long and narrow. All these shapes will depend upon the function they perform. The shape of the cell may vary with the function they perform. Is it clear? Size depend on the location, shape depend on the function. Largest isolated single cell is the egg of ostrich, and in our multicellular organism, RBC is this much in diameter. Nerve cell is the longest cell. They can vary in shape. Different shapes are there: disc-like, polygonal, columnar, cuboidal, thread-like, even irregular. The thread-like, you have to remember, it's a nerve cell. They can ask you which is branched and long to nerve cell. All this they can ask you. From here, questions are there, beta. Is it clear? The shape depend upon the function what they perform. Size, you do one thing, right? Size here in one box area. All the size, typical bacteria, bacteria cell, mycoplasma, PPLO, typical eukaryotic cell, virus, every size you write down, RBC size. Okay, now this much is clear. Now I'm starting with the prokaryotic cell. The prokaryotic cell means bacteria, cyanobacteria, mycoplasma, and PPLO. They are generally smaller and multiply more rapidly than the eukaryotic cell. They also vary greatly in shape and size. We have studied bacteria shapes. Total 4 shapes were there: coccus, bacillus, comma, and spirulum. So they also vary shapes. 4 basic shapes we have already studied of the bacteria. If you see, it is similar even through prokaryotes. All the prokaryotes are having same organization, though they are different in the shape and function. All prokaryote cells are having a cell wall surrounding the cell membrane except mycoplasma. So we have already studied in biological classification. Mycoplasma is wall-less monera. It is wall-less monera. We have already studied it. Fluid-filled matrix of the cell is known as cytoplasm. You know, prokaryotes don't have nucleus. Genetic material is naked. Naked means what? It means it is not enveloped by nuclear membrane. They are having which DNA? Genomic DNA. Genomic DNA, single chromosome is circular DNA. So ploidy of bacteria is haploid. That is why we say bacteria ploidy is haploid. Question can be asked because we are mentioning single chromosome, and single chromosome is itself known as circular DNA. So in addition to this, leave this is already present. They are having many bacteria have a small circular DNA outside the genomic DNA, and these are known as plasmid. So genomic DNA is one, but plasmid can be one to many. That is why if you remember in the biotech, we told that plasmid is used as a vector. These plasmid DNA give special property, unique phenotype character to the bacteria like resistance to antibiotic. And because of this property, we have used it as a vector for cloning the genes. And if you remember bit in the biotech chapter, if suppose recombinant DNA is given to the bacteria, and bacteria, if it has taken it, we will say bacteria is transformed. So DNA, this plasmid DNA is used to monitor bacterial transformation. with foreign DNA, whether the bacteria has taken the recombinant DNA or not. Is it clear? Prokaryotes have something unique in the form of inclusion bodies. Inclusion bodies are also present. They are having differentiated form of cell membrane called mesosome, and this is a characteristic of prokaryotes. Prokaryotes are having this only thing. Mesosomes and inclusion bodies, these are the things which are present only in prokaryotes. Now, cell wall and cell membrane. Glycocalyx, if it is loose, it is known as slime layer. If it is thick, it is known as capsule. Cell wall is made up of peptidoglycan. Because of differential in staining, we will say if they are able to retain the color, we will say gram positive or gram negative, depend upon the difference in the response to the staining procedure which is developed by Christian Gram. So difference in the staining in the cell wall, we are saying they are gram positive or gram negative. Cell membrane, this is the structure which is similar to eukaryote. Similar to eukaryote, and again it was PMT question. Because just like in eukaryotes, their cell membrane is also made up of phospholipid bilayer and protein. Now cell membrane modification. Cell membrane modification. First is mesosome. They are making mesosomes. The mesosome exists it exists as tubule, lamella, and vesicles. Second modification is chromatophore. Now, how to learn the mesosome function? See, very easy presentation. Imagine this is a diagram of bacteria, I'm drawing it. This is I'm drawing mesosome only right now. So this is cell membrane I've drawn. This is cell wall. And this is your outermost glycocalyx. Clear, beta? And now you do one thing, draw this way, a single circular DNA, bacterial DNA known as genomic DNA. We say bacteria are very simple in structure because of this reason. Now, if you see the mesosome is holding the DNA, it is helping in DNA replication. Equal distribution to two daughter cells. After replication, equal distribution. If you don't see the DNA, isn't it resembling like a cristae? Finger-like projections in mitochondria. And you know, cristae are responsible for respiration, so mesosome help in respiration. Majority of the ribosomes are attached to the nuclear membrane. So these, plasma membrane. So here the ribosomes are attached. Though they are present free in the cytoplasm, but they are attached. So if ribosomes are there, it means they are doing the protein synthesis. Protein means enzyme, and few enzymes secretion purpose also. Is it clear? So this is the function of the mesosome. From here we can make 7 functions. DNA replication, equal distribution to daughter cells, septa wall formation, respiration, enzymatic, secretion. Is it clear? And holding the DNA in the place. Chromatophore, chroma word is coming to it is itself telling it is having the pigment. So it is present in which prokaryote? It will be present in cyanobacteria. Cyanobacteria, it will be present where they are containing the pigment, and this pigment which they are having the pigment, that is chlorophyll A, and they are having phycobilins. Phycobilins can be of 2 types, phycocyanin making it blue color, phycoerythrin making it red color. Generally, when they come on the surface, they are developing more phycocyanin. That is why we say blue-green algae. Clear? Now let us see the NCERT. See carefully, beta. Most prokaryotic cell, especially the bacterial cell, they have chemically complex cell envelope. Cell envelope consists of tight bound 3 layered structure. First is your glycocalyx, then is cell wall, and then is cell membrane. Now they are performing, each layer is performing, see carefully, each layer will perform distinct function, but together they act as a single protective unit. Now, depending upon this, they are staining procedure. They are classifying bacteria into two categories, gram positive and gram negative. Which retain the color, we say gram positive, which loses the color, we say gram negative. Now we are discussing one by one, glycocalyx. It differs in composition and thickness also. If it is loose, it is known as slime layer. If it is thick and tough, it is known as capsule. So I think so glycocalyx is clear. If it is thick, it is known as capsule. If it is loose, known as slime layer. Next is cell wall. Cell wall, if you see, it will determine the shape of the bacteria and protect bacteria from bursting. Because bacteria generally faces the hypotonic condition. In hypotonic condition, they prevent bacteria from bursting, and in hypertonic condition, they prevent bacteria from collapsing. Then third is cell membrane. Cell membrane is this membrane is similarly structured to that of eukaryote. This is the question I told you, PMT question. This structure is similar in prokaryotes and eukaryotes. Now, membranous structure first is your mesosome. Mesosome, it can exist in three forms: vesicles, tubule, lamella. This only I wrote it in the notes. They help in focus function. Number 1, cell wall formation. Number 2, DNA replication. Number 3, distribution to daughter cells. Number 4, respiration, secretion, increase the surface area. Yes, this function. And enzymatic content. So it is helping in all the 7 functions. Please note down. In some prokaryotes like cyanobacteria, there is another membranous extension known as chromatophore, which contain pigment. The chromatophore contain pigment. So I think so cell envelope and its modification are clear. Now see the bacteria flagella. See the diagram better. On this you are finding some structures are also attached. See, this is your membranous structure. Let me draw the diagram. Imagine this is one rod-like structure which is present here inside it. In this, rings will be present. So this is making basal body. This is hook and this is filament. So this part is making basal body. This is known as hook and this is known as filament. Filament, this is a structure of bacterial flagella. The bacterial flagella is made up of which protein, beta? Flagellin protein. It is made up of flagellin protein. Please remember, eukaryotic flagella is made up of tubulin protein, but this is made up of flagellin protein. Apart from this, it is having one more structure. See carefully, I'll draw this way. This is known as, just for understanding, I'm drawing it. Please remember, this is pili. Pili is long. This is pili. Pili is made up of pillin protein. Another structure, I'm drawing it. This is fimbriae, this way. Fimbriae, and fimbriae is bristle-like. Pili diagram is telling you tubular, fimbriae diagram is telling you bristle-like. Now see the NCERT. Bacteria can be motile or non-motile. If motile, they are having very thin filamentous extension from their cell wall, called flagella. Please focus, they can frame the question from cell wall to cell membrane. Bacteria shows range in the number and arrangement. Why range in number? If one flagella, you see monotrichous. This way arrangement, you will say amphitrichous. This way arrangement, you will say cephalotrichous, lophotrichous, or you can say peritrichous. Arrangement and number can change. Bacteria flagella has 3 parts: basal body, hook, filament. Filament is the longest portion and extend out from the cell surface. Beside this, they are having 2 more structure. They are also surface structure, but do not play the role in motility. One is your pili. Pili is elongated tubular structure, tube-like structure made up of a special protein that is known as pillin protein. Fimbriae are small, bristle-like. They are helping in adhesion. They help bacteria in adhesion. They help bacteria to attach to the rock or stream or whatever it is. So Fimbriae helps in adhesion, villi helps in conjugation. So I think so this much is clear. Now see ribosome. If you remember, I have drawn the diagram, I focus that majority of the ribosomes are bound on the cell membrane. So in prokaryotes, the ribosomes are associated with cell membrane. That is why we say, now size, please remember 15 to 20 nanometer and 2 subunit 50 and 30s and together 70s. So draw this way ribosome. This is 30s. Don't apply math here. Ki ma'am, math total is coming 80. No, together is 70s. And do this one right here, 15 nanometer by 20 nanometer. So you will learn the size. You know very well, they are the site for protein synthesis. And many ribosomes, see, this is one messenger RNA on which many ribosomes are attached. Messenger RNA, single messenger RNA on which many ribosomes are attached. So we say polyribosome or polysome. The single messenger RNA where several ribosomes are attached to form a chain known as polysome or polyribosomes, which is helping in translating. You know, ribosomes are protein factories, so they are converting the messenger RNA language into the language of the protein. Now, inclusion bodies. Reserve food material in prokaryote cells are stored in the form of inclusion bodies. The inclusion bodies will store food and they are not bounded by any membrane system. Please, they are not bounded by any membrane system. But when we say gas vacuole, people think it is surrounded by membrane. But this membrane is not true cell membrane. It is membrane made up of only protein. So see carefully, very important. Please try to focus here now. First is your gas vacuole I'm discussing. Gas vacuole are found in blue, green algae and purple and green photosynthetic bacteria. So you write down here, it is found in cyanobacteria, it is found in green sulfur bacteria, it is found in purple sulfur bacteria. And if you remember, we have already studied green sulfur and purple sulfur word in the photosynthesis chapter. So gas vacuole is present in these three. Please remember. Why? Because cyanobacteria are doing first oxygenic photosynthesis. So they are storing the oxygen and as a result, they are able to come from the bottom of the ocean on the surface. So first, this is clear. Now next, important. I want to focus the difference between the cyanophacian granules and glycogen granules. See, bacteria will store glycogen. The glycogen granules is made by bacteria. Glycogen granules are made by bacteria and cyanophacian granules are made by cyanobacteria, means blue-green algae, made by blue-green algae. And if you see this bacteria, glycogen granule, because they are storing the glycogen, that is why glycogen granule. But cyanobacteria is storing cyanophacian starch. It stores cyanophacian starch. Cyanophacian will store starch. Please remember, bacteria will store glycogen. Cyanobacteria will store starch. Next inclusion body is your phosphate granules. So that is common. That is, you have to just learn that they are phosphate granules. So total, how many inclusion bodies you have learned, beta? Number one is your phosphate granule you should learn. Number two, cyanophacian granules. Number three, glycogen granules. And number four, gas vacuole. These all are your inclusion bodies. So just do one thing, just go through. If you have any doubt in prokaryotic cell, then ask me.